Hello dear students, welcome. Today we are going to learn Chapter 5, The Life Processes, from Standard 10 Biology Science subject. I hope this video will become helpful for you, so let's begin the journey. How do we differentiate between living and non-living? 1. Observing life. We often identify living beings by movement, e.g., dog running, man shouting. Even during sleep or rest, signs like breathing indicate life. Movement is a commonly used sign but not sufficient to define life. 2. Plants and life. Many identify plants as alive due to their green color or growth. However, not all plants are green, and not all show visible growth at all times. Conclusion. Growth and visible movement are not always reliable indicators of life. 3. Invisible movements. Molecular movement, e.g., movement of proteins, enzymes, etc., happens inside cells. These cannot be seen with the naked eye but are essential for life. 4. Are viruses alive? Viruses do not show molecular movement unless inside a host. This is why there is controversy about whether viruses are truly alive. 5. Why molecular movement is important. Living organisms are highly organized structures: cells, tissues, organs. Environmental effects tend to break down this order. Organisms must constantly repair and maintain themselves to stay alive. This maintenance requires continuous molecular activity. Conclusion. Visible movement alone is not a sufficient sign of life. Internal molecular activity is a key indicator of life. Life is maintained by repair, growth, molecular transport, and organization. Chapter 6. Life Processes. Section 5.1. What are life processes? Definition. Life processes. The basic processes that are essential for an organism to maintain life and carry out body functions, even when at rest, e.g., sitting or sleeping. Why life processes are necessary? To maintain and repair the body's structure. To prevent breakdown due to environmental effects. Require a constant supply of energy. Sources of energy. Energy comes from food, which must be taken in nutrition, broken down respiration, to release usable energy. Nutrition. The process of transferring energy and raw materials from food, external source, into the body. Food is mainly carbon-based. Different organisms use different types of nutritional processes, depending on the complexity of food. Respiration. A process to break down food using oxygen in most organisms, to release energy. Involves oxidation reduction, redox reactions. Converts food into a uniform source of energy for molecular movements and growth. Simple versus complex organisms. Single-celled organisms. All processes occur via direct contact with environment. No need for specialized systems. Multicellular organisms. Not all cells are in contact with the environment. Need specialized tissues and organ systems for nutrition, gas exchange, respiration, transportation, excretion, transportation. Required because food and oxygen are taken up at one place but needed by all cells. A transport system moves food and oxygen to cells. Waste products away from cells. Excretion. Chemical reactions in the body produce waste byproducts. Some are harmful and must be removed. Excretion is the process of removing waste from the body. In complex organisms, this involves specialized excretory tissues. Waste is transported to these tissues by the transport system. Summary of life processes. Life process function. Nutrition intake and breakdown of food for energy. Respiration release of energy from food using oxygen. Transportation distribution of food, oxygen and removal of waste. Excretion removal of harmful metabolic waste. Section 5.2. Nutrition. Definition of nutrition. Nutrition. The process of taking in food and converting it into energy and materials required for growth, repair, and maintenance of the body. Need for nutrition. Provides energy for physical activities, e.g., walking, cycling. Supplies raw materials for growth, development, synthesis of proteins and other biomolecules. Maintaining the internal order of the body, homeostasis. Types of nutrition. Type description. Autotrophic organisms prepare their own food using inorganic substances, CO2 and H2O. Heterotrophic organisms depend on other organisms for food. Autotrophic nutrition. Organisms use carbon dioxide and water to prepare food. Requires chlorophyll, sunlight, performed mainly by green plants, some bacteria e.g. cyanobacteria. Heterotrophic nutrition. Organisms depend on complex organic substances from other organisms. These substances must be broken down into simpler forms before use. Enzymes, biocatalysts, help in breaking down complex food into simple, absorbable forms. All animals and fungi are heterotrophs. Key differences between autotrophs and heterotrophs. Feature autotrophs, heterotrophs. Food source inorganic, CO2 plus H2O. Organic, complex food substances. Dependency self-sufficient, dependent on autotrophs. Examples green plants, algae, some bacteria, animals, fungi, some bacteria. Food preparation through photosynthesis or chemosynthesis through ingestion and digestion. Role of enzymes. Enzymes are biocatalysts. They speed up biochemical reactions, especially in digestion and metabolism. Essential for breaking complex food into simpler molecules usable by cells. Summary points. All living organisms require energy and raw materials from food. Autotrophs make their own food. Heterotrophs depend on others. Enzymes are essential for converting complex food into usable forms. Ultimately, heterotrophs depend on autotrophs directly or indirectly. Section 5.2.1. Autotrophic nutrition. What is autotrophic nutrition? A mode of nutrition where organisms prepare their own food from simple inorganic substances like CO2 and H2O using sunlight. Process used. Photosynthesis. Photosynthesis. Definition. Process by which green plants and some bacteria use sunlight, carbon dioxide, and water to produce carbohydrates, glucose. Chlorophyll is essential to capture sunlight. Photosynthesis reaction. H2 plus HO. With help of chlorophyll, in presence of sunlight, makes glucose plus release O2. Steps/events of photosynthesis. Absorption of light energy by chlorophyll. Conversion of light energy into chemical energy and splitting of water into hydrogen and oxygen. Reduction of carbon dioxide to form carbohydrates. These steps may not occur simultaneously. Example. Desert plants take in CO2 at night and store it for use during the day. Storage of food in plants. Glucose produced during photosynthesis is used for energy. Stored as starch in plants. In humans, excess glucose is stored as glycogen in the liver and muscles. Role of chloroplasts. Photosynthesis takes place inside chloroplasts. Chloroplasts are green-colored cell organelles found in leaf cells. They contain the pigment chlorophyll, which absorbs sunlight. Importance of chlorophyll. Essential for capturing solar energy. Experiment shows that without chlorophyll, photosynthesis does not occur, demonstrated through variegated leaf activity. Summary table. Component role in photosynthesis. Carbon dioxide CO2. Raw material from air. Water H2O. Raw material from soil. Sunlight energy source. Chlorophyll absorbs sunlight. Present in chloroplasts. End product glucose used or stored. Oxygen released. Autotrophic nutrition. Detailed notes, pointwise. 1. Definition. Autotrophic nutrition is a mode of nutrition in which organisms prepare their own food from simple inorganic substances like carbon dioxide and water using an external energy source, usually sunlight. The word auto means self, and trophic means nutrition. 2. Organisms showing autotrophic nutrition. Green plants, algae, Some bacteria, e.g. cyanobacteria. These organisms are called autotrophs. 3. Raw materials required for autotrophic nutrition. Raw material source and role. Carbon dioxide, CO2. Taken from air through stomata in leaves. Water, CHO. Absorbed from soil through roots. Sunlight captured by chlorophyll, energy source. Chlorophyll, green pigment that absorbs light energy. 4. Site of photosynthesis. Takes place in the chloroplasts of leaf cells. Chloroplasts contain chlorophyll, the green pigment that traps sunlight. 5. The process of photosynthesis. Photosynthesis is the biochemical process used by autotrophs to prepare food. Chemical equation. 6CO2 plus H2O in presence of sunlight, and with the help of chlorophyll plant make C6H12O6 plus 6O2. Where CO2 equals carbon dioxide. CHO equals water. 6O6 equals glucose. O2 equals oxygen. 6. Major events in photosynthesis. Absorption of light energy by chlorophyll. Conversion of light energy into chemical energy. Splitting of water molecules into hydrogen and oxygen. Photolysis. Reduction of CO2 to form carbohydrates. Glucose. 7. Storage of food. Immediate use. Glucose is used by plant cells to release energy. Storage. Excess glucose is converted into starch and stored in leaves, roots, seeds, stems. In humans, glucose is stored as glycogen. 8. Importance of chlorophyll. Acts as a solar energy trapper. Converts light energy to chemical energy. Essential for photosynthesis. Without it, photosynthesis does not occur. 9. Adaptations in desert plants. In dry environments, plants, e.g. cacti, open stomata at night to absorb CO2. CO2 is stored as an intermediate compound and used during the day when sunlight is available. This adaptation prevents excessive water loss. 10. Significance of autotrophic nutrition. Basis of the food chain. Provides energy and organic material for all life forms. Produces oxygen as a byproduct which is essential for respiration in aerobic organisms. Maintains the balance of CO2 and O2 in the atmosphere. 11. Comparison with heterotrophic nutrition. Feature autotrophic, heterotrophic. Food source inorganic, CO2 plus H2O. Organic, from other organisms. Dependency independent, dependent on autotrophs. Mode of nutrition photosynthesis or chemosynthesis, ingestion followed by digestion. Examples: plants, algae, cyanobacteria, animals, fungi, many bacteria. 12. Experimental proof. Variegated leaf experiment. Shows that chlorophyll is essential for photosynthesis. When exposed to sunlight and tested with iodine, only the green parts of the leaf turn blue-black, proving starch, food, is formed only where chlorophyll is present. Summary. Autotrophic nutrition enables certain organisms to produce their own food using sunlight, CO2, and H2O. Photosynthesis is the core process of autotrophic nutrition. It is essential for the survival of all living organisms and maintains the atmospheric balance of gases. How do plants obtain carbon dioxide? Detailed notes. 1. Role of carbon dioxide in photosynthesis. Carbon dioxide, CO2, is one of the essential raw materials required for photosynthesis. It is absorbed from the atmosphere. 2. Entry of CO2 through stomata. Stomata are tiny pores present mostly on the lower surface of leaves. They allow gaseous exchange, entry of CO2 and exit of O2 and water vapor. Each stomatal pore is surrounded by 2 kidney-shaped guard cells. 3. Structure and location of stomata. Found on the epidermis, outer surface, of leaves' main site. Also present to a lesser extent on stems and young roots. Figure 5.3, NCERT, shows a labeled stomatal structure. 4. Function of stomata. Allows intake of CO2 for photosynthesis. Release of O2 as a byproduct. Loss of water vapor during transpiration. 5. Opening and closing mechanism. Controlled by guard cells. When guard cells absorb water, become turgid, stomata open. When guard cells lose water, become flaccid, stomata close. 6. Water regulation. Since water is also lost through open stomata, plants regulate the opening based on time of day, usually open in day, closed at night. Availability of water. Photosynthetic need. Closed when so not needed. 7. Other sites of gaseous exchange. Though leaves are primary, gaseous exchange can also occur through stems, especially green stems. Roots, via diffusion through soil-air spaces. Summary table. Feature description. Structure stomata, pore, plus guard cells. Function gas exchange, CO2 in, O2 out, plus water loss. Control guard cells regulate opening/closing. Importance essential for photosynthesis. How do plants get the requirements for photosynthesis? Detailed point-wise explanation. 1. Basic requirements of photosynthesis. To carry out photosynthesis, plants need the following: carbon dioxide, CO2, water, HO, sunlight, chlorophyll. 2. How plants obtain carbon dioxide, CO2. Source: atmosphere. Enters through stomata, tiny pores, mainly on the underside of leaves. Mechanism: gaseous exchange happens when stomata open. Guard cells control the opening and closing of stomata. O2 diffuses into leaf cells where photosynthesis occurs. 3. How plants obtain water, H2O. Source: soil. Absorbed by root hairs through the process of osmosis. Transported via xylem tissue. Reaches leaves where it is used in the splitting reaction, photolysis, during photosynthesis. 4. How plants obtain sunlight. Source: captured by chlorophyll, green pigment, in chloroplasts of leaf cells. Function: absorbs light energy. Converts it into chemical energy required for photosynthesis. 5. Role of chlorophyll. Located in chloroplasts inside leaf cells. Function: traps solar energy. Initiates the light-dependent reactions of photosynthesis. Without chlorophyll, photosynthesis cannot occur. 6. Summary of how plants get each requirement. Requirement source / means role in photosynthesis. Carbon dioxide atmosphere of stomata provides carbon for glucose, C6H6. Water soil roots xylem, leaves source of hydrogen and oxygen from splitting. Sunlight sun captured by chlorophyll energy for driving the reaction. Chlorophyll present in chloroplasts in leaf cells captures light energy. Final notes. All these components work together to enable the plant to prepare its own food in the form of glucose. Excess food is stored as starch. Oxygen produced is released into the air as a byproduct. Stomata. Structure, function and role. For Class 10, Life Processes Chapter 1. What are stomata? Stomata are tiny pores or openings present mainly on the lower surface of leaves. They help in gas exchange and transpiration in plants. 2. Structure of stomata. Each stoma, singular of stomata, consists of stomatal pore, central opening through which gases and water vapor move. 2. Guard cells, bean-shaped cells, kidney-shaped in dicots, dumbbell-shaped in monocots, that control the opening and closing of the pore. Chloroplasts. Guard cells contain chloroplasts and perform photosynthesis. Subsidiary cells surround guard cells, not always present. 3. Function of stomata. Function explanation. 1. Gaseous exchange allows CO2 to enter for photosynthesis and O2 to exit as byproduct. 2. Transpiration releases water vapor during daytime which helps in cooling the plant. 3. Maintains water balance, controls water loss by regulating opening/closing based on plant need. 4. Photosynthesis regulation opens during the day to allow CO2 entry for photosynthesis. 4. Mechanism of opening and closing. Controlled by turgor pressure in guard cells. When water enters guard cells, they become turgid stomata open. When water exits guard cells, they become flaccid stomata close. Influenced by light, humidity, CO2 concentration, internal water level of the plant. 5. Role of stomata in photosynthesis. Allows CO2 to enter leaf tissues for photosynthesis. Removes O2 produced during the process. Indirectly controls the rate of photosynthesis by regulating CO2 availability. Summary table. Feature description. Location lower epidermis of leaves, also on stems and young roots. Main parts stomatal pore, guard cells, chloroplasts. Functions gas exchange, transpiration, controls water loss. Controlled by turgor pressure in guard cells. Significance essential for photosynthesis and water regulation. Location of stomata, pointwise for easy learning. Main location. Lower surface of leaves. Especially in dicot plants like rose, mustard. Also found on upper surface of leaves in monocots like grass, where stomata are usually present equally on both surfaces. Green stems G in xerophytes like cacti. Herbaceous parts, non-woody stems. Young roots, rare in some aquatic or aerial plants. Not found on old stems and roots, which are woody and covered by bark. Flowers, fruits, seeds. Usually do not have stomata. Key fact for exams. Most abundant on the underside of dicot leaves. Equal distribution on both sides of monocot leaves. Raw materials required by autotrophs, besides energy. Pointwise notes, NCERT-based. 1. Water. Source. Absorbed from the soil by roots. Function. Used in photosynthesis and transport of nutrients. Also helps maintain turgidity in plant cells. 2. Mineral nutrients. Plants require several inorganic minerals from the soil to build body tissues and perform life processes. 3. Important minerals and their roles. Mineral role in plants form absorbed from soil. Nitrogen N, needed for synthesis of proteins, nucleic acids, and chlorophyll nitrates, NO3. Nitrites, NO2, or organic nitrogen compounds. Phosphorus, P, important for ATP, DNA, and cell membrane formation phosphates, PO4 superscript 3. Iron Fe, required for chlorophyll synthesis and enzyme activation ferrous, Fe superscript 2. Ferric, Fe superscript 3, forms. Magnesium, G, central atom in chlorophyll molecule, activates enzymes Mg superscript 2. 4. Nitrogen source, 2 ways. Inorganic, directly absorbed from soil as nitrates or nitrites. Organic, formed from atmospheric nitrogen by nitrogen-fixing bacteria, e.g., rhizobium in legume roots. 5. Summary. Plants need more than just carbon dioxide and water. They require mineral nutrients for cell growth, protein and enzyme synthesis, DNA/RNA formation, energy transfers like ATP. Heterotrophic nutrition. Definition. Heterotrophic nutrition is the mode of nutrition in which organisms depend on other organisms, plants or animals, for their food because they cannot prepare their own food. These organisms consume complex organic substances which are broken down into simpler forms using enzymes. Key features. No photosynthesis. Energy and nutrients are obtained from external sources, plants/animals. Require enzymes to digest complex food molecules. Found in animals, fungi, and many bacteria. Types of heterotrophic nutrition. Type description examples. 1. Holozoic types of organisms. Ingest solid food, digests it, and absorbs nutrients and removes waste, for example. Human beings, amoeba, cow. 2. Saprophytic organisms will obtain food from dead and decaying organic matter, for example fungi, e.g., bread mold, bacteria. 3. Parasitic organisms, lives on/in a host and derives nutrients from it, often harming the host, for example. Cuscuta, a bell, lice, tapeworm. 4. Symbiotic type organism, two organisms live together and benefit each other nutritionally, for example. Lichen, alga plus fungus, rhizobium in legume roots. Examples of heterotrophic organisms. Group example type of nutrition. Animal human lion holozoic. Protozoa amoeba, Paramecium holozoic. Fungi mushroom, yeast saprophytic. Plants Cuscuta, a bell, parasitic. Bacteria Lactobacillus saprophytic. Comparison. Autotrophs versus heterotrophs. Feature autotrophs, heterotrophs. Food source make own food, photosynthesis, depend on others. Chlorophyll present absent. Examples green plants, algae animals, fungi, many bacteria. 5.2.3. How do organisms obtain their nutrition? 1. Variation in feeding mechanisms. Nutrition depends on organism complexity and food type. Simple organisms use their entire body surface for food intake. Complex organisms have specialized organs/systems for digestion and absorption. Unicellular organisms. Examples. Amoeba. Mode. Holozoic nutrition. Food is engulfed using pseudopodia, temporary finger-like projections. Forms a food vacuole inside the cell. Digestion occurs inside food vacuole using enzymes. Complex food is broken into simple substances. Nutrients diffuse into cytoplasm. Undigested waste is ejected out from the cell surface. Key term. Pseudopodia equals false feet, used for both movement and food capture. Paramecium. Also a unicellular organism but with defined shape. Nutrition also holozoic. Cilia, tiny hair-like structures, help to sweep food particles towards a specific oral groove, mouth-like spot. Digestion occurs in food vacuoles, similar to amoeba. Summary. Comparison between amoeba and paramecium. Feature amoeba paramecium. Body shape irregular definite. Food intake anywhere on surface, pseudopodia. Specific oral spot, cilia aid. Locomotion pseudopodia cilia. Food vacuole present. Nutrition in amoeba. Step-by-step explanation. Amoeba shows a holozoic mode of nutrition, which includes ingestion, digestion, absorption, assimilation, egestion. Step 1. Ingestion, phagocytosis. Amoeba senses the presence of food, like bacteria, algae, and extends pseudopodia, false feet, toward it. The pseudopodia surround and engulf the food particle, enclosing it in a food vacuole. This process of engulfing solid food particles is known as phagocytosis. Step 2. Digestion. Inside the food vacuole, digestive enzymes are secreted. These enzymes break down complex food substances into simpler soluble forms like glucose, amino acids, etc. Step 3. Absorption. The digested nutrients from the food vacuole diffuse into the cytoplasm of the amoeba. These nutrients are used for energy and cellular functions. Step 4. Assimilation. Absorbed nutrients are utilized for growth, repair, and energy production through respiration. It helps in the formation of new cytoplasm and organelles. Step 5. Egestion. The undigested waste remains in the food vacuole. It moves to the cell membrane and is expelled out by rupturing the surface. Dear students, next topic in digestive system in human being. Let's begin. Definition of digestion. Digestion is the process of breaking down complex food substances, like carbohydrates, proteins, and fats, into simpler, absorbable forms using mechanical and chemical means. Digestive system in humans. Overview. Type of nutrition, holozoic. Involves 5 basic steps. Ingestion, intake of food. Digestion, breakdown of food. Absorption, nutrient uptake into blood. Assimilation, utilization by cells. Egestion, removal of waste. Functions of digestive system. breaks down food into absorbable molecules, provides energy and raw materials for body growth and repair, removes undigested waste. 1. Mouth or oral cavity. We begin with the mouth, located in the head region. This is where digestion starts when we take food in, called ingestion. The teeth play a vital role here by chewing and breaking food into smaller pieces. This is mechanical digestion. Meanwhile, saliva from the salivary glands moistens the food and begins chemical digestion of starch with the help of an enzyme called salivary amylase. 2. Pharynx. Next is the pharynx, located behind the mouth in the throat region. It acts as a common passage for both food and air and connects the mouth to the esophagus. 3. Esophagus, food pipe. The food now enters the esophagus or food pipe, which runs through the neck and chest region. Here, rhythmic muscle contractions called peristalsis push the food down into the stomach. For stomach. Now the food enters the stomach, a muscular sac located in the upper left part of the abdomen. Here, food is mixed and churned further. More mechanical digestion. The stomach secretes hydrochloric acid, HCl, which kills harmful microbes and provides an acidic medium for the enzyme pepsin to start protein digestion. Mucus is also secreted to protect the inner lining from the acid. 5. Small intestine, duodenum, jejunum, ileum. After the stomach, food moves into the small intestine, which is divided into 3 parts, duodenum, jejunum, and ileum, and occupies the central abdominal region. This is the main site of digestion and absorption. It receives bile from the liver to emulsify fats, and pancreatic juices to digest carbohydrates, proteins, and fats. Nutrients are absorbed into the blood through the intestinal walls. 6. Liver. The liver, located in the upper right abdomen, plays a major role in digestion by producing bile, which helps break down fats into smaller droplets. Bile is stored in the gallbladder. 7. Gallbladder. The gallbladder, situated just beneath the liver, stores and concentrates bile, and releases it into the duodenum when needed for fat digestion. 8. Pancreas. The pancreas, located below the stomach, is a gland that secretes pancreatic juice into the small intestine. This juice contains vital enzymes, amylase for carbohydrates, trypsin for proteins, and lipase for fats. 9. Large intestine. Any undigested food now enters the large intestine, which surrounds the small intestine. Its primary job is to absorb water and minerals, and the remaining waste is compacted into feces. 10. Rectum. The rectum, at the end of the large intestine, stores feces temporarily until it is ready to be eliminated. Anus. Finally, feces are expelled from the body through the anus in a process called egestion. So, in summary, the human digestive system involves mechanical and chemical digestion, absorption of nutrients, and elimination of waste, with each organ playing a specialized role. Isn't it amazing how our body is designed to handle such a complex process? Human digestion, detailed notes. Definition of digestion. Digestion is the process by which large, complex food molecules are broken down into smaller, absorbable units, like glucose, amino acids, fatty acids, and glycerol, by digestive enzymes by mechanical and chemical means, to be absorbed and used by the body. Digestive system overview. The human digestive system consists of alimentary canal, digestive tract. 1. Mouth, oral cavity. 2. Pharynx. 3. Esophagus, for stomach. 5. Small intestine with its division, duodenum, jejunum, ileum. 6. Large intestine with its 3 divisions, cecum, colon, rectum. And finally, 7. Anus. B. Digestive glands. Salivary glands. Parotid, submandibular, sublingual. Liver, produces bile. Pancreas. Produces digestive enzymes and bicarbonate. Gastric glands, in stomach wall. Intestinal glands. In small intestine. Digestion in the mouth. In detail. Step by step. From ingestion to bolus formation. 1. Ingestion, eating food. The process starts when food enters the mouth, also known as the oral cavity. The food may be solid, semi-solid, or liquid. 2. Structures in the mouth and their roles. Structure role. Teeth will mechanically break down food like chewing or mastication into smaller pieces to increase surface area. The tongue will help in mixing food with saliva, tasting, and pushing it toward the pharynx. Salivary glands will secrete saliva, which contains enzymes and mucus for lubrication and chemical digestion. Palate, hard and soft, will separates the oral cavity from the nasal cavity, aids in swallowing. At last, uvula will prevents food from entering the nasal passage during swallowing. 3. Saliva and its composition. Saliva is secreted by 3 major pairs of salivary glands. Parotid, near ear. Submandibular, under jaw. Sublingual, under tongue. Components of saliva. Water 99.5%. Salivary amylase, ptyalin. Starts digestion of starch. Mucus. Lubricates the food. Lysozyme. Antibacterial enzyme. Electrolytes. Maintain pH. 6.8. 7.2. 4. Chemical digestion in the mouth. Salivary amylase begins the breakdown of starch, polysaccharide, into maltose, disaccharide. This is the first step of chemical digestion. Reaction. Starch converted into maltose by salivary amylase enzyme. But remember that no digestion of proteins or fats occurs in the mouth. 5. Mechanical digestion, chewing. Teeth cut and grind the food into smaller pieces. Tongue rolls and mixes it with saliva. This process is called mastication. 6. Formation of bolus. The thoroughly chewed and saliva-mixed food becomes soft, slippery, and rounded. This semi-solid mass is called the bolus. The bolus is easier to swallow and is pushed to the pharynx by the tongue. 7. Swallowing deglutition. A reflex action that pushes the bolus from the mouth to the pharynx, and then into the esophagus. The epiglottis closes the windpipe, trachea, during swallowing to prevent choking. Digestion in the stomach. Detailed notes. Food enters the stomach. After forming bolus, the food is swallowed, deglutition, and moves through the pharynx into the esophagus. Movement through the esophagus is aided by peristalsis, wave-like muscular contractions. The lower esophageal sphincter, also called cardiac sphincter, opens to allow the bolus into the stomach and then closes to prevent backflow, acid reflux. Sphincter valves. Gatekeepers of food flow. Sphincters are muscular valves that control the entry and exit of food within different parts of the digestive tract. 1. Lower esophageal sphincter, cardiac sphincter. Located between the esophagus and the stomach, near the heart. Prevents acid reflux, backflow of stomach acid into esophagus. Opens when bolus arrives from the esophagus. Closes after food enters the stomach to maintain one-way movement. 2. Pyloric sphincter. Located between the stomach and the duodenum, first part of small intestine. Controls release of chyme from the stomach into the small intestine. Opens gradually after digestion in the stomach is complete. Stomach. Location, structure and function. Location. Located in the upper left side of the abdominal cavity, just below the diaphragm. Lies between the esophagus and small intestine, duodenum. Structure of the stomach. The stomach is a J-shaped muscular organ with 4 major regions. Cardiac region, where food enters from esophagus. Fundus. Dome-shaped upper portion. Body main central region, major site of digestion. Pyloric region, connects to the duodenum, ends in pyloric sphincter. Layers of the stomach wall. Layer function. Mucosa, inner. Layer secretes gastric juice. Contains gastric glands. Submucosa layer, connective tissue with blood vessels and nerves. Muscularis externa layer has 3 muscle layers. Longitudinal, circular and oblique. Helps in churning food. Serosa, outer. Layer protective layer. Gastric glands and gastric juice. Gastric glands are located in inner layer called mucosa, contain 3 types of cells, which secrete digestive enzymes. Cell type secretion function. Mucus cells. Mucus protects stomach lining from acid. Peptic, chief cells. Pepsinogen, inactive enzyme, converts into pepsin to digest proteins. Parietal, oxyntic, cells. HCl and intrinsic factor. HCl activates pepsinogen. Intrinsic factor helps absorb vitamin B2. Composition of gastric juice. Water. 1. HCl, hydrochloric acid. It kills bacteria and make food acidic by changing pH. 2. Pepsinogen, inactive enzyme, which activated in acidic pH under the effect of HCl and digests protein from food. 3. Gastric lipase. For mucus. It will line inner wall of stomach and protect it from HCl effect. 5. Intrinsic factor, for vitamin B2 absorption. Functions of gastric juice components. Component function. Kills microbes. Activates pepsinogen, pepsin. Provides acidic pH. 1.5. 2.0. 4. Enzyme activity. Pepsinogen, pepsin. Pepsin digests proteins, peptide. Gastric lipase, minor digestion of fats, especially in infants. Mucus, protects the inner lining from acid and enzyme. Intrinsic factor, helps in absorption of vitamin B2 in ilia. Rugae, folding magic of the stomach. Rugae are folds in the mucosal lining of the empty stomach. They expand and flatten as the stomach fills with food. Allow the stomach to stretch up to 1.5 liters without tearing. Also help in gripping and churning the food. Food retention in stomach. Food remains in the stomach for about 3 to 4 hours. During this time it is mixed with gastric juice, churned and partially digested. This process is both mechanical, churning, and chemical, enzyme activity. What happens inside the stomach? Mechanical digestion. Muscular walls churn and mix food with gastric juice. This helps in uniform breakdown of food particles. Chemical digestion. Pepsin begins digestion of proteins into short peptides. Very little digestion of fats, gastric lipase. No carbohydrate digestion here. Chyme formation. Semi-digested mixture. After 3 hours, food becomes semi-liquid, acidic, and partially digested called chyme. Chyme contains proteins partially digested into peptides. Fats minimally digested. No carbohydrate digestion. Starch digestion stops due to acidic pH. Chyme is released in small amounts into the duodenum via the pyloric sphincter for further digestion. After 3 to 4 purs of digestion in stomach we get chyme, which further digested into small intestine. So let's begin. Chyme in duodenum and mixing with bile and pancreatic juice. Entry of chyme into duodenum. After 3 to 4 hours in the stomach, the partially digested acidic food, chyme, is slowly released into the duodenum, the first part of the small intestine. This release is regulated by the pyloric sphincter. Neutralization of chyme. Chyme from the stomach is highly acidic, Pit2. As it enters the duodenum, it is neutralized by bicarbonate ions, HCO3, secreted by the pancreas, making the environment suitable for enzyme activity in the small intestine. Bile. Secretion from liver, via gallbladder. What is bile? Bile is a yellowish-green alkaline fluid secreted by the liver and stored in the gallbladder. It contains no enzymes, but plays a key role in fat digestion. Composition of bile. Bile salts, e.g., sodium terephthalate. Bile pigments, bilirubin and biliverdin. Cholesterol. Electrolytes. Water. Function of bile. Emulsification of fats. Breaks large fat globules into smaller droplets, increasing surface area for lipase action. Alkaline nature. Helps neutralize acidic chyme. Aids in absorption of fat-soluble vitamins. A, D, E, K. Pancreatic juice. Secretion from pancreas. Composition of pancreatic juice. Secreted by the exocrine pancreas, it contains bicarbonate, HO3. Neutralizes acid. Digestive enzymes. Pancreatic amylase. Digests carbohydrates, starch into maltose. Trypsinogen, inactive. Activated to trypsin digest, proteins into peptides. Chymotrypsinogen convert to chymotrypsin and digest, proteins into peptides. Procarboxypeptidase converted into carboxypeptidase digest, peptides into amino acids. Pancreatic lipase. Digests fats into fatty acids and glycerol. Nucleases. Digest DNA and RNA. Activation of protein digestive enzymes. Enterokinase from intestinal mucosa. Converts trypsinogen to trypsin. Trypsin then activates other protein-digesting enzymes. Chymotrypsinogen, procarboxypeptidase. Mixing in duodenum. Coordinated action. The bile duct and pancreatic duct open into the duodenum at the common hepatopancreatic duct, guarded by the sphincter of Oddi. Chyme, bile, and pancreatic juice mix in the duodenum to continue digestion. This marks the beginning of complete enzymatic digestion of proteins, carbohydrates, and lipids. See for yourself, read and remember it. Once chyme has been mixed with bile and pancreatic juice in the duodenum, the process of digestion continues in the remaining parts of the small intestine, the jejunum and ileum. 1. Structure of the small intestine. The small intestine is divided into 3 parts. First, the duodenum, where digestion begins with the help of bile and pancreatic juice. Next, the jejunum, the middle part, where most of the digestion and nutrient absorption takes place. And finally, the ileum, which completes the process of nutrient absorption. 2. Intestinal juice. Succus entericus. The walls of the small intestine have glands that secrete intestinal juice, also known as succus entericus. This fluid is slightly alkaline, with a pH around 7.8, and contains mucus, bicarbonates, and several digestive enzymes. 3. Enzymes and their functions. Let's look at the enzymes found in intestinal juice and what they do. Maltase breaks maltose into 2 glucose molecules. Lactase acts on lactose to give glucose and galactose. Sucrase splits sucrose into glucose and fructose. Peptidases convert peptides into amino acids. Lipase continues fat digestion into fatty acids and glycerol, especially after bile emulsification. Nucleotidases break down DNA and RNA into nitrogen bases and sugars. 4. Final digestion and nutrient absorption. After all this enzyme action, food is broken down into its simplest absorbable forms. Monosaccharides like glucose, amino acids, fatty acids and glycerol, along with vitamins and minerals. These nutrients are then absorbed through the walls of the intestine. 5. Villi and absorption. The inner lining of the small intestine is covered with villi, and each villus is lined with even smaller microvilli. Together, they form a brush border. This structure increases the surface area enormously, allowing maximum absorption of nutrients. Glucose and amino acids are absorbed into blood capillaries and carried to the liver through the hepatic portal vein. Fatty acids and glycerol are absorbed into lacteals, the lymph vessels inside villi, and then transported through the lymphatic system into the bloodstream. 6. Movements in the small intestine. To help in mixing and pushing the food forward, 2 main types of movements take place in the small intestine. Segmentation, which involves rhythmic contractions to mix food with enzymes, and peristalsis, a wave-like motion that pushes the digested content forward. Food stays in the small intestine for around 4 to 5 hours for complete digestion and absorption. 7. Digestion in the large intestine. After absorption is complete, undigested food passes into the large intestine. The large intestine includes the cecum, colon, rectum, and ends at the anus. No enzyme secretion here. Unlike the small intestine, the large intestine does not secrete digestive enzymes, and no further digestion takes place here. 9. Functions of the large intestine. But it has other important jobs. It absorbs water and electrolytes, like sodium and chloride. It forms semi-solid feces from undigested food. It secretes mucus to ease the movement of waste. And most interestingly, it houses gut bacteria, which help in synthesizing vitamin K and B-complex vitamins, and ferment remaining carbohydrates. 10. Feces formation and defecation. The final waste is now formed into feces, which is stored in the rectum. When the time is right, the defecation reflex is triggered, and the feces are passed out through the anus. 11. Summary. To sum up, the jejunum and ileum complete digestion and absorb nutrients. The villi and microvilli make absorption highly efficient. And the large intestine reclaims water, forms feces, and supports vitamin production by gut flora. Next topic is respiration in organisms. Let's understand respiration in organisms step by step, in a simple pointwise format. 1. Purpose of respiration. After we eat food, the energy stored in glucose needs to be released for our body to function. This process happens inside our cells and is called respiration. The energy released is used for all life activities, like growth, repair, movement, and cell maintenance. This energy is stored in a special molecule called ATP, the energy currency of the cell. First step, glycolysis. The first step of respiration is glycolysis. Here, one molecule of glucose, which has 6 carbon atoms, is broken down into 2 molecules of pyruvate, each with 3 carbon atoms. This happens in the cytoplasm of the cell and occurs in both aerobic and anaerobic respiration. 3. Fate of pyruvate. 3 possible pathways. Now, the pyruvate formed can follow 3 different pathways depending on the availability of oxygen. A. Aerobic respiration. With oxygen. This is the most efficient pathway and takes place inside the mitochondria. It occurs in most plants, animals, and humans. The pyruvate is completely broken down into carbon dioxide, water, and a large amount of energy, about 38 ATP molecules. Equation. C6H2O6 plus 6O2, 6O2 plus 6H2O plus energy. ATP. This is why aerobic organisms can perform high-energy activities. B. Anaerobic respiration. Without oxygen in yeast. In organisms like yeast, where oxygen is not available, pyruvate is converted into ethanol, carbon dioxide, and a small amount of energy, only 2 ATP molecules. This process is called fermentation and is used in bread making and alcohol production. C. Anaerobic respiration in human muscles. Temporary. During heavy exercise, our muscles may not get enough oxygen. So, the pyruvate gets converted into lactic acid and a little energy. This lactic acid builds up in the muscles and causes cramps and fatigue. Later, when oxygen becomes available again, lactic acid is removed. 4. ATP, the energy currency. The full form of ATP is adenosine triphosphate. This molecule stores energy in its phosphate bonds. When needed, ATP is broken down into ADP and Pi, releasing energy. This energy is used in muscle movement, protein synthesis, nerve signals, and active transport in cells. 5. Comparison. Aerobic versus anaerobic respiration. Let's quickly compare the 2 types. Feature aerobic respiration, anaerobic respiration. Oxygen required. No. Site mitochondria cytoplasm. End product CO2 plus H2O ethanol plus CO2 / lactic acid. ATP produced around 38 per glucose, only 2 per glucose. Found in humans, animals, plants, yeast, some bacteria, human muscles. 6. Real-life applications. Fermentation by yeast is used to make bread soft and fluffy, and to brew alcoholic beverages. Muscle cramps after intense exercise are due to lactic acid buildup, a result of anaerobic respiration. Exchange of gases in plants and animals. Detailed notes. 1. Need for oxygen in aerobic organisms. Aerobic respiration requires oxygen to break down food molecules, glucose, for energy. Thus, organisms must have a mechanism to intake oxygen and eliminate carbon dioxide. 2. Gaseous exchange in plants. Plants do not have specialized respiratory organs like animals. Gases are exchanged through stomata, pores on leaf surface, lenticels in stems, root hairs in roots. How gases are exchanged in plants occurs via simple diffusion, movement of gases from high to low concentration. Intercellular spaces inside leaves allow easy movement of gases to all cells. Diffusion in plants depends on environmental conditions, time of day, plants' metabolic activity, 3 day versus night in plant gas exchange, time photosynthesis respiration main gas released, day yes active, yes oxygen, night no yes carbon dioxide, daytime. CO2 produced during respiration is used in photosynthesis. So, oxygen is the major gas released. Nighttime. No photosynthesis occurs. Only respiration happens, CO2 is released into the air. 4. Gaseous exchange in animals. Animals need to take in oxygen and expel carbon dioxide from the body. Unlike plants, animals have specialized respiratory organs. Types of respiratory organs. Habitat example organisms respiratory organs. Terrestrial humans lions lungs. Aquatic fish, tadpoles gills, extract O2 from water. Amphibians frogs lungs plus moist skin. Insects grasshopper tracheal system. 5. Aquatic versus terrestrial respiration. Terrestrial animals breathe atmospheric oxygen through lungs or tracheae. Aquatic animals use oxygen dissolved in water via gills. Water contains less oxygen than air, hence, aquatic organisms breathe faster or more efficiently. Key differences. Plant versus animal respiration. Feature plants, animals. Organ for gas exchange stomata, lenticels, roots lungs, gills, tracheae. Exchange method simple diffusion, specialized structures and circulation. Gases involved O2 and CO2, O2 and CO2. Active during day/night respiration. Always. Photosynthesis. Day only continuous respiration. 5.3.2. Respiratory system in human beings and other organisms. Aquatic versus terrestrial respiration. Aquatic organisms, e.g. fish. Oxygen is obtained from dissolved oxygen in water. Water is taken in through the mouth and passed over the gills. Gills extract dissolved oxygen transferred to blood. Rate of breathing is much faster in aquatic animals because less oxygen is available in water compared to air. Terrestrial organisms, e.g. humans, dogs, use atmospheric oxygen for respiration. Oxygen is absorbed through specialized organs. Lungs in humans/mammals. Tracheae in insects. Skin in earthworms, amphibians. B. Key features of respiratory organs. In terrestrial organisms. All respiratory organs have a large surface area for efficient gas exchange. The exchange surface is thin and delicate to allow gases to diffuse easily. The surface is located inside the body to protect it. There are passageways, like nostrils, trachea, to carry air to and from the respiratory surface. A mechanism, muscles/diaphragm, is required to move air in and out. See human respiratory system. Step by step. 1. Entry of air. Air enters through nostrils. It is filtered by fine hairs, moistened and cleaned by mucus, warmed before reaching lungs. 2. Passage of air. Passes through nasal passage, pharynx, throat, larynx, voice box, trachea, windpipe supported by cartilage rings to prevent collapse. 3. Inside the lungs. Trachea divides into bronchi. Each enters one lung. Bronchi divide into bronchioles. Bronchioles end in alveoli air sacs. 4. Alveoli, site of gas exchange. Tiny balloon-like structures at the end of bronchioles. Large surface area for maximum diffusion. Surrounded by blood capillaries. Gas exchange. Oxygen diffuses into blood. Carbon dioxide diffuses into alveoli from blood. 5. Breathing mechanics. Action. What happens? Result. Inhalation. Diaphragm contracts and flattens. Ribs lift up. Chest cavity expands. Air sucked in. Exhalation. Diaphragm relaxes. Ribs fall down. Chest cavity contracts. Air pushed out. 6. Residual volume. Even after exhalation, some air remains in lungs. Ensures continuous exchange of gases. D. Transport of gases in humans. 1. Oxygen transport. Carried by hemoglobin in RBCs, red blood corpuscles. Hemoglobin has high affinity for oxygen. Forms oxyhemoglobin in lungs, releases oxygen in tissues. 2. Carbon dioxide transport. More soluble in water than oxygen. Carried in dissolved form in blood plasma. A small amount is also carried by hemoglobin as carbaminohemoglobin. Summary points. Aquatic organisms respire faster due to low oxygen in water. Human respiratory system includes nostrils, trachea, bronchi, lungs, alveoli. Alveoli are the site of gas exchange. Breathing involves muscular movement, diaphragm and ribs. Oxygen is carried by hemoglobin, and CO2 is mostly carried in dissolved form. Let's understand how we breathe. The mechanism of breathing in humans. Step 1. Inhalation. Breathing in. When we inhale, the diaphragm contracts and moves downward, and the rib cage expands upward and outward. This increases the space inside the chest cavity. As a result, the air pressure inside the lungs becomes lower than the outside air pressure. So air naturally rushes into our lungs through the nose. It travels from the nostrils, pharynx, larynx, trachea, bronchi, bronchioles, and finally reaches the alveoli, the tiny air sacs in the lungs. Step 2. Exchange of gases in alveoli. Inside the alveoli, a very important exchange happens. Oxygen from the inhaled air diffuses into the blood, while carbon dioxide from the blood moves into the alveoli to be exhaled. This exchange is possible because alveoli have thin walls and rich blood supply, allowing gases to easily pass in and out. Step 3. Exhalation, breathing out. Now it's time to exhale. The diaphragm relaxes and moves back up, and the rib cage comes down and inwards. This reduces the space in the chest cavity, increasing the pressure inside the lungs. As a result, the air containing carbon dioxide is pushed out through the same pathway, from alveoli to nostrils. Step 4. Continuous cycle. This cycle of inhalation and exhalation happens about 12 to 20 times per minute, and it's vital, because it provides our cells with oxygen and removes the waste gas carbon dioxide. So next time you take a breath, remember, there's a lot happening behind the scenes to keep you alive and healthy. 5.4.1 Transportation in human beings. Q1. Why is transport system needed? Multicellular organisms, like humans, have millions of cells far from the external environment. Cells need oxygen, nutrients, and removal of wastes. A proper transport system is essential. Simple diffusion is too slow for large organisms. E.g., it would take 3 years for oxygen to diffuse from lungs to toes without blood and hemoglobin. Blood. The transport medium. Blood is a fluid connective tissue. Functions. Transports food, gases, hormones, waste. Components. Plasma, 55% liquid part, transports food, CO2, nitrogenous wastes, salts, etc. Red blood corpuscles. RBCs transport oxygen using hemoglobin. White blood cells. WBCs fight infections. Platelets. Help in blood clotting. Transported by plasma. Nutrients. Glucose, amino acids, vitamins. CO2 in dissolved form. Nitrogenous wastes, urea, uric acid, hormones, mineral salts, transported by RBCs. Oxygen binds to hemoglobin, forms oxyhemoglobin. Hemoglobin has a high affinity for oxygen. CO2 is less bound to RBCs, mostly dissolved in plasma. Did you know? Total surface area of alveoli equals 80 m², like a tennis court. This large area maximizes gas exchange efficiency. Hemoglobin speeds up oxygen transport drastically. B. Human circulatory system. Main components. Heart, the pumping organ. Blood, transport medium. Blood vessels, arteries, veins, capillaries. Heart, structure and function, brief preview. Feature details. Chambers 4. Right atrium, right ventricle, left atrium, left ventricle. Valves prevent backflow of blood. Double circulation, blood passes twice through heart in one cycle. Oxygenated blood from lungs, heart, body. Deoxygenated blood from body, heart, lungs. Detailed structure and working will be covered in the next subsection. Questions from NCERT text, answer key. Q1. Advantage of terrestrial organisms over aquatic ones. Air has more oxygen than water. Terrestrial organisms breathe more efficiently due to easy oxygen availability. Diffusion of oxygen from air is faster than from water. Q2. Ways of oxidizing glucose for energy. Organism type condition end product. Yeast anaerobic ethanol plus CO2. Human muscle cells temporary anaerobic lactic acid. All cells, usually. Aerobic CO2 plus H2O plus energy, ATP. Q3. Oxygen and CO2 transport in humans. Oxygen binds with hemoglobin in RBCs, transported as oxyhemoglobin. O2 carried dissolved in plasma, 70%, some as carbaminohemoglobin. Q4. Lung design maximizing gas exchange. Lungs contain millions of alveoli. Alveoli have large surface area, 80 m², thin walls, rich blood supply, enables efficient diffusion of oxygen and carbon dioxide. Activity 5.7. Investigation questions. Use this for real-world biology project or field activity. You're asked to investigate normal hemoglobin range in humans. Compare values for children versus adults, men versus women. Veterinary data for buffalo, cow, calf, males versus females. You should record values. Compare patterns. Discuss why differences exist, e.g., menstrual cycle, muscle mass, age. Explanation. Structure of the human heart, pointwise. Location and shape. The heart is located in the thoracic cavity, between the lungs, slightly to the left side of the chest. It is cone-shaped and roughly the size of your clenched fist. Protective covering. It is enclosed in a double-layered membrane called the pericardium, which protects the heart and reduces friction during beating. Between the layers is pericardial fluid that acts as a lubricant. Heart wall. The heart wall is made of 3 layers. Epicardium, outermost thin layer. Myocardium, thick, muscular middle layer, responsible for contraction. Endocardium, smooth inner lining. Septum, dividing wall. The heart is divided vertically by 2 septa, walls. Interatrial septum, separates right and left atria, upper chambers. Interventricular septum, separates right and left ventricles, lower chambers. So, the heart is divided into left and right halves. Chambers of the heart. Each half has 2 chambers. Upper chambers, called atria, singular, atrium. Lower chambers, called ventricles. So in total, 4 chambers, right atrium, right ventricle, left atrium, left ventricle. Walls of atria and ventricles. Atria have thin walls because they only receive blood and push it to the ventricles, short distance. Ventricles have thicker muscular walls because they pump blood out of the heart. Left ventricle has the thickest wall because it pumps blood to the entire body. Right ventricle pumps blood only to the lungs, so it is less thick than the left. Valves of the heart to prevent backflow. Tricuspid valve. Between right atrium and right ventricle. Bicuspid, mitral, valve. Between left atrium and left ventricle. Pulmonary valve. At the opening of pulmonary artery from right ventricle. Aortic valve. At the opening of aorta from left ventricle. Major blood vessels connected to the heart. Vena cava, superior and inferior, brings deoxygenated blood from body to right atrium. Pulmonary artery. Carries deoxygenated blood from right ventricle to lungs. Pulmonary veins. Bring oxygenated blood from lungs to left atrium. Aorta. Carries oxygenated blood from left ventricle to the whole body. Internal flow of blood. Double circulation. Deoxygenated blood. Body right atrium, right ventricle, lungs. Oxygenated blood. Lungs, left atrium, left ventricle, body. Double circulation. Explained. What is double circulation? Double circulation means that blood passes through the heart twice during one complete cycle of circulation in the body. This is a characteristic feature of birds and mammals, including humans. Why is it called double? Because the heart pumps blood in two separate circuits. Pulmonary circulation, between the heart and lungs. Systemic circulation, between the heart and rest of the body. Pathway of double circulation. 1. Pulmonary circulation, right side of the heart. Deoxygenated blood from the body enters right atrium. Moves into the right ventricle. Pumped to the lungs via the pulmonary artery. In lungs, blood releases CO2 and absorbs O2. Now it becomes oxygenated blood. 2. Systemic circulation, left side of the heart. Oxygenated blood from the lungs enters left atrium. Moves into the left ventricle. Pumped into the aorta. Carried to all parts of the body. Body uses oxygen and nutrients. Blood becomes deoxygenated again. Cycle repeats. This deoxygenated blood then returns to the right atrium, and the cycle repeats. Importance of double circulation. Keeps oxygen-rich and oxygen-poor blood separated. Ensures high-pressure blood flow to body organs. Improves efficiency of oxygen and nutrient delivery. Valves of the heart ensure unidirectional flow. Atrioventricular AV valves. Tricuspid valve. Between right atrium and right ventricle. Bicuspid, mitral, valve. Between left atrium and left ventricle. Semilunar valves. Pulmonary valve. Between right ventricle and pulmonary artery. Aortic valve. Between left ventricle and aorta. Flow of blood through the heart, double circulation. Body right atrium, right ventricle, lungs, for oxygenation. Lungs, left atrium, left ventricle, body to supply oxygen. Fun facts. Heartbeat 72 times/min. Pumps 5 liters of blood/min. Controlled by sinoatrial, say, node. The natural pacemaker. Blood vessels. Explained, teacher style, pointwise. What are blood vessels? Blood vessels are tubular structures that carry blood throughout the body. They are part of the circulatory system and help in the transport of oxygen, nutrients, hormones, and waste products. Types of blood vessels. 1. Arteries. Carry oxygenated blood away from the heart to body parts. Exception. Pulmonary artery carries deoxygenated blood to lungs. Have thick, muscular, and elastic walls to handle high pressure. Blood flows in pulses due to heartbeats. No valves. 2 veins. Carry deoxygenated blood towards the heart. Exception. Pulmonary vein carries oxygenated blood from lungs. Have thin, less muscular walls and larger lumen. Blood flows under low pressure, so valves are present to prevent backflow. Blood flow is steady, not pulsed. 3. Capillaries. Microscopic, thin-walled vessels, 1 cell thick. Connect arterioles, small arteries, to venules, small veins. Allow exchange of gases, nutrients, and wastes between blood and body cells. Extremely numerous and present in all tissues. Flow summary. Arteries, arterioles, capillaries, venules, veins. Let's now compare the three types of blood vessels: arteries, veins, and capillaries, in a simple table format. Ready? Let's begin. One direction of flow. Arteries carry blood away from the heart. Veins carry blood toward the heart. Capillaries are tiny vessels that connect arteries and veins. Two types of blood. Arteries mostly carry oxygenated blood. Veins mostly carry deoxygenated blood. Capillaries are the exchange points, where oxygen and nutrients leave the blood, and carbon dioxide and waste enter the blood. 3-wall thickness. Arteries have thick and elastic walls to withstand high pressure. Veins have thin walls, as the pressure is much lower. Capillaries have walls that are only 1 cell thick to allow easy exchange of substances. For valves, arteries do not have valves. The blood flows under high pressure. Veins have valves to prevent backflow of blood. Capillaries also have no valves. 5 pressure. Blood flows in arteries under high pressure. In veins it flows under low pressure. In capillaries the pressure is moderate, just enough for exchange to happen smoothly. So, arteries are strong and fast-flowing, veins return blood slowly with valves, and capillaries are thin exchange bridges between the two. That's the beautifully coordinated system of blood vessels in our body. Lymphatic system. Explained. Definition. The lymphatic system is a part of the circulatory and immune systems. It consists of lymph vessels, lymph nodes, and lymphoid organs. Its main job is to return excess tissue fluid to the blood and defend the body against infections. Location. It is spread throughout the body just like the blood vessels. Lymphatic vessels are found in almost all body tissues, except the brain and spinal cord. Lymph nodes are located in neck, armpits, chest, abdomen, and groin. Major organs include spleen, thymus, tonsils, and bone marrow. Structure. Lymph. A colorless fluid similar to plasma but without RBCs. Contains WBCs, mostly lymphocytes, proteins, lipids, and waste. Lymphatic vessels. Thin-walled tubes that carry lymph. Begin as tiny lymph capillaries in tissues. Merge into larger lymphatic ducts, which return lymph to the bloodstream. Lymph nodes. Small bean-shaped filters located along lymph vessels. Contain immune cells, lymphocytes, macrophages, that filter pathogens. Lymphoid organs. Tonsils, in throat. Fight inhaled infections. Thymus. Behind sternum. Matures T lymphocytes. Spleen. Stores and filters blood, destroys old RBCs. Bone marrow. Produces all blood cells including lymphocytes. Functions of lymphatic system. Drainage of excess fluid. Collects excess tissue fluid and returns it to the bloodstream, maintaining fluid balance. Fat absorption. In the small intestine, lacteals, special lymph vessels, absorb fats and transport them to blood. Immune defense. Lymph nodes and lymphoid organs help to detect and destroy pathogens like bacteria and viruses. Transport of white blood cells. Lymph transports lymphocytes and other immune cells throughout the body. Summary table. Component description. Lymph clear fluid with WBCs. Lymph vessels carry lymph from tissues to veins. Lymph nodes filter lymph and fight infection. Thymus matures T cells, type of lymphocyte. Spleen filters blood, stores WBCs. Lacteals absorb fats from the intestine.